Hi guys, welcome to our second inflammation video. During the first inflammation video, we talked about what inflammation hopes to achieve, what the primary goals of inflammation are, and the four cardinal signs of inflammation, as well as the mobilization of the defenses that are associated with inflammation. So next we're going to talk about the second priority and the most important priority of inflammation, which is the containment and destruction of pathogens. So again, that's the containment and destruction of pathogens will prevent pathogens from spreading throughout the body. So this could be because of trauma, where they've gained a point of entry, or if they're already in the body and they've already entered and they're starting to cause trouble. So when there's acute trauma, fibrinogen filters into tissue fluid clots, and that can help form a sticky mesh that walls off microbes. This can occur without having observable trauma, but there could be some localized trauma that you don't really see. So it doesn't necessarily mean that there's been a break in the skin. Heparin also helps with the containment and destruction of pathogens because it helps prevent clotting at the site of injury. So that's the pathogens are then going to be they're going to be in a fluid pocket that's surrounded by the clot, and there they can be attacked by antibodies, phagocytic cells, and other defenses so that they can be contained and destroyed. Neutrophils, if you remember, are granulocytic leukocytes, and these are the main antibacterial cells within the body, so that they can accumulate at the site of injury within an hour. And after leaving the bloodstream, they exhibit chemotaxis. If you remember, chemotaxis is the ability to attract other chemicals that are generally pro-inflammatory so that it is a positive feedback loop. So these chemicals can be different chemicals such as bradykinin and the leukotrienes that guide other chemicals, other pro-inflammatory chemicals to that injury site. So neutrophils are quick responders to bacteria, and they're aggressively antibacterial. That's because they're able to phagocytize the cells. They also have what's called a respiratory burst, which means that they create antioxidant chemicals that cause the bacteria to burst. They also secrete cytokines to recruit macrophages and more neutrophils. So when that happens, the macrophages and T cells will secrete colony stimulating factors that stimulate leukopoiesis, which is the production of more leukocytes, and that then raises the white blood cell count in blood. So if you remember, if you got differential white blood cell count, you could see if there's different leukocytes that are represented in the blood outside of normal. So remember again, you would get neutrophilia, which is an increase in neutrophils during bacterial infection, or you could get eosinophilia. That would be an elevated eosinophil count with allergy or a parasitic, usually helminthic worm infection. So during tissue cleanup and repair, which is the third stage of inflammation, monocytes are the primary agents of tissue cleanup and repair. So remember that monocytes leave the red bone marrow, and then they become macrophages when they leave the blood and go into the tissue. So they arrive to the site of injury in about 8 to 12 hours, and then they mature into macrophages, and they're able to engulf and destroy bacteria, the damaged host cells, and dead and dying neutrophils. So basically, they just eat up everything that needs to go away. So edema also contributes to the tissue cleanup, so that swelling compresses veins and will reduce venous drainage. It also forces open the valves of the lymphatic capillaries, promoting lymphatic drainage. Remember that the lymphatic capillaries generally rely on pressure for their flow through them. The lymphatics are able to collect and remove bacteria, dead cells, proteins that are no longer needed, as well as tissue debris, better than blood capillaries are able to. So pus is the yellow accumulation, or white, or whatever color, of dead neutrophils, bacteria, cellular debris, and tissue fluid. If something has a lot of pus. It is not pussy. It is purulent, by the way. And so sometimes you can get abscesses, which are an accumulation of pus within a tissue cavity. And usually those will need antibiotics or will need some sort of drainage to get rid of them. So platelet-derived growth factor is secreted by blood platelets and endothelial cells of the vessels in that injured area. And this will help with repair. So this will stimulate fibroblasts to multiply, which will help with the repair mechanisms. And it also causes collagen to be synthesized. Hyperemia, which means an increase in blood flow that's usually associated with inflammation, will allow for an excess of oxygen, amino acids, and other things that are needed for protein synthesis. So again, that redness, swelling, and heat does have a function. So hyperemia is able to do quite a few different things during the whole inflammation process. So also that increased heat, that's one of the 4 cardinal signs of inflammation, increases metabolic rate, will speed up mitosis, and can speed up tissue repair. So it's kind of cool because metabolic rate during certain circumstances, like during fever, can actually raise up to 25%, which is kind of insane. These are very metabolically costly. So fibrin clots, if you remember from our clotting lecture, are able to form a cloth or a scaffold for tissue reconstruction, so it makes it a little bit easier. And pain also is able to limit the use of that body part, so it has a chance to rest